Hi there. Welcome to the Genomics module. So, genomics is a field in which we study the entire DNA sequence or the genome of an organism. So, genome is the entire DNA sequence which include your mRNA, proteins, and other things like this. So, in this module, we delve into various aspects of your genomics analysis, exploring the topics like references genomes, working with the gene sequence, how to find gene ontologies, how to find gene annotations, and how to annotate a genome and extract some information from it. So, first of all, we will see to work with high quality reference genome. So, the high quality reference genome that I chose is Plasmodium falciparum genome. So, this section provides the insights into GC content distribution across the Plasmodium falciparum genome. So researchers can analyze the JC distribution pattern to understand the various genomic features. So what I am talking about is I'm talking about your code section. So here you can see if I move to the first section of the genome, we will see working with high quality reference genome. So what we do here. So in this code, we analyze the GC content of a genome and then try to visualize it. So first we will deal with the presentation file. So here we will see we will process and analyze the genomic data and visualize the genome. Like what genome mainly consists of? Genome mainly consists of chromosomes. So we will analyze how much chromosomes are present in your genome. So our genome of interest is Plasmodium. So and we also analyze the GC content and then identify some genomic features and some insights into your genome. So this code also generate a PNG image for visualizing the genome as your chromosome location. So this visualization help researchers to visualize genome organization and gain a better understanding of the genome structure. So first, what we will see here. So we will import some necessary libraries. After importing some libraries, we will define the name of the file. So I'm going with the Plasmodium genome. Then we will read the genome sequence of the file. After that, we will iterate over the genome sequence and print their description. Then we will import the library sectels, which deals with the sequences. And then we will initialize the dictionaries to store chromosome size and GC content. Then we will read the sequence again and then iterate over the genomic sequence. And then we will check if the sequence represent any chromosome and extract the chromosome number from the description of the genome. And then again, we will iterate over the sequence blocks in the defined size and calculate the GC content of the block and then print the minimum and maximum content which is present in the genome. So if we see here you can see we have this whole kind of output that is presented here. And here we can see there is a maximum and minimum GC to be found. So here you can see here is the minimum and here is the maximum GC content of the whole genome. And after that, we will extract the chromosome numbers and how much chromosomes are present there. We will extract it from a genome file. And then we will find the largest chromosome and then initialize the variable of color grading the chromosome based on GC content. So then we will iterate over the chromosome and create chromosomal representation. And after that, we will iterate over the GC content blocks and color them accordingly. And after that, we will add the chromosome representation of the genome and then draw the chromosome and save it as a image, which is PNG image. So here you can see these are the chromosomes. So here is the chromosome number 1, 2, 3, 4, 5, 6, 7, 8, 9, 2 up to 14. So all the blue color is showing the GC content which is present there and other kind of the chromosome contain low number of GC content, then there is low bluish, that if you have more amount of GC content then it will be red in this. So after that, what we can look into. So we can analyze the gene sequence. we can extract gene sequence, retrieve specific gene or retrieve specific sequence from your FASTA genome file, and extract the coding sequence from the region of sequence of the gene and translate them into protein as well. So study protein translation of specific gene to identify its function is impactful on an organism. So what I'm trying to say is, if we have this uh, if we can extract a feature from the genome, for example, let's just say if we want to extract a protein, if we want to extract a gene from the uh, chromosome and then change the gene to a protein and translate it or transcribe it. So first of all, what we have to do, we have to import the modules and then define the UniProt server, because we are obtaining the sequence from UniProt, and after that we will send the request to fetch the human gene P53, and we will be retrieving the limited columns of 15 traits related to P53. And note, we might revisit the CAG database in the future to see the functional impact as well. Then we will convert the response to a data frame using pandas. I already told you about panda library, and get the UniProt entry from the human P53 and then sort. And after getting the entry point, we will get the a record of P53 human using Express database, and then print some information about UniProt, and then print the comments and keyword associated with that gene or P53 human protein, and get a list of unique features that is identified there. And after getting a list, we will cross check this in our UniProt record, express A, and then print them. And then we will get a list of unique gene ontology annotation in the UniProt record and then print them. So here you can see I got these entries, which is protein coding gene starting from this and uh, this starting from this location and ending at this location. This is the positive strand that is presented on, and this is the ID that is given by the gene. We can also identify different other genes, but I just chose these. And then we can also getting a voltage gated sodium channel, which is the gene ontology of this gene. So this is how you can extract feature of gene using a database. And if we move on to another slide, we can annotate a genome. We can count the number of features present in the GFF file and GFF database and list them. Also, we can retrieve all the unique IDs and categorize them by feature type, count the number of mRNA or exon features, and identify additional information which is present in the genome. We can also analyze the genomic data of the gene and gather specific information like the number of exons per gene, genes with the maximum number of exon, minimum number of exon, total number of genes present in the database or the genome. So if we move on to our core section, we will just import the libraries. We will define the URL there the GFF file is present, and then we will connect to the gffutils, which is a library for connecting to a database, and we will print the list of features that is present in our GFF file, count the number of features and each feature type in the database, get the unique IDs, and after that initialize the default dicts to count the number of mRNAs and exons of each region, then loop through each sequence ID and analyze the genes in that region, then calculate the span of the gene difference between start position, end position, and which strand it is present on, then get the mRNA for the gene and count the mRNAs of all gene, determine if the gene has exons or not, if the exon check is either gone or the mRNA feature is present there, get the exon feature and count the number of exons of each gene. After that, we will print the gene with the maximum number of exon and its count, and then we will print the gene with the maximum span and its span value, and then we will print the counts of mRNA and exons of each gene. So here you can see here is the gene description that we were talking about. Is it a mRNA? It is have an exon. If it has a 5' UTR region or 3' UTR region. So this is basically the annotation of the genome. So if you just click here, you can see it is present in the text format. So here you can see all the values that are present here. You can predict them according to your own needs, and you just have to change the file name like this. So the GFF file that you have, you just have to change your GFF file name, and then everything should be same and your analysis should be up and running. Then we will move on to working with low quality reference genome. For example, if you have a reference genome that doesn't contain that high quality data, it is not refined enough, you can also get some valuable information from it. So that means we can analyze the contigs, we can calculate the maximum length of contigs, we can visualize the fraction of contigs and calculate the sequence statics. We can get the various fraction of n contigs, which is, and then plot them accordingly to your own needs. So let's just move on to code. And here we will import the libraries, download the genome. If you want to download, you can download the genome and then name it here, and then display the descriptions, calculate the number of your ends that was contigs in your chromosome, and then display the fraction of them, and then calculate the statics which is written in the code. And after that, create a plot showing the fraction of contigs in different sizes in your genome file. So that's pretty much it for your low fraction genome if you want to run it. And then we can also find orthologs of your gene. If you are familiar with the paralogs or orthologs concept, you should know about that. So species data, it consists of three major information. Species data, obtain the relevant species data through your ensemble REST API request. So this is the first module that we deal with when we are going to find a ortholog using the ensemble API. Then we will find the information about gene transcript. In fact, the information regarding available transcript data for your specified gene and retrieve across references to external databases. Then we will fetch the information regarding the ortholog gene and print the details about them, such as their species, taxonomy level, how their ensemble ID, their gene ontology, and things like this. So if we move on to the code section, here you can see we have to import the ensemble server and then just send the get request to check out in our genes. To check if the request was successful, raise an exception, get information about the species. Here you can enter your species name, get information about the external databases, look up for the information of the LCT gene, which is present in the Homo sapiens humans, and get the DNA sequence of your gene, get the cross reference again with the specified ensemble ID, and then check the gene with the specified ensemble ID and external database, which is Go is the gene ontology database. Get the homology information of the gene from the ensemble ID and type ortholog and low sequence data. Retrieve the list of homologs that you want to find and move on to getting the information of the homologs gene in horse using your ensemble ID. I just search for horse in LCT gene. My query was LCT gene and I searched it in the horse genus and I got the homologs gene of horse as well. Here you are seeing that uh, these are all the information that was present here. So if you can scroll on that, the DNA sequence was already coming through and all the homologs that were orthologs, that was identified are present here. So discussing the gene ontology. So this code features the gene ontology annotations of lactase gene that retrieve your Go terms. So what is gene ontology basically? Gene ontology is kind of function of your gene. So in the bioinformatics term, you get these specific gene functions that are associated with your ontologies, And then the code provides a hierarchical representation of your term with your genes. And it is easier to do Go annotation using the Shiny Go and different databases, but you can also get this by using the code. So here is the code. We have to just make sure you request your server and then prepare the parameter of the request, specify the ensemble ID, which is the LCT lactase gene, and then cross over for the specify ensemble ID, then extract the gene ontology and retrieve ontology information of that ID. So that's pretty much it. Here you can get all the information. So retrieve the information about specific Go ID. You can also do that as well. If you know the Go ID, if I can just show you what it to do. So if we move on to our web browser and go to the Go database, which is a gene ontology resource, you can search. You should know that there are specific Go IDs that are given to specific functions. So this will be associated with the lactase activity can be found in the following location. So this is the detailed information. Here you can see detail annotation. So it is a lactase activity, molecular function, lactose degranulation activity, catalysis. There is degalactose, galactose, and things like this. So then we will retrieve the information of the parent term. Then we will function that parent term and then get the data of our ID. So all this is just for getting the functional IDs related to your tree, related to your term. So if we have this ID, which is a molecular function lactase activity, it is the catalysis of the reaction that we were just seeing there. Here we were seeing the catalysis of the reaction, lactose, degalactose, and this. And it is the same activities that is representing here. These are all the molecular functions that are associated with the lactase LCT gene that we were looking at. So that was the main whole phenomena of working with genomics. I know this could be quite not understanding and things like that, but the thing you have to do, these kind of codes are now provided everywhere. So you just have to download these code and just know what they are trying to do with this code and just input your file and use the code. You don't have to write all this code all over again. Just make some changes, spring some salt in it, and then go on with it. And then concluding this module provide you valuable insight into genomic analysis, exploring references genome, gene sequence, genome annotation, gene ontologies, gene annotations, and once again, essential to interpret and analyze genomics data. Also, you can unlock the significant insight into the genome, how you can explore your genome, what is GFF file. You should learn all this by yourself if you want to. By exploring the genome, various organisms, you can better understand different species, how they are related to orthologs, how homologs are related, and you can also achieve some great advancement of medicine, environmental science, agriculture, and ultimately it leads to better understanding of how living functions, your living organisms function on this planet. Sorry for that. So this source comprises us that we can use the BioPython and use the Python code to analyze the genomes as well. We can see how much chromosomes are present there in a genome. We can do that by ourselves. So thank you so much for listening to this, and we will see you in the next.